Madam, you may keep your camera turned off now.
ಶುರು ಮಾಡೋಣ ಮೇಡಮ್?
ಹಾ ಸರ್.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಇಂದಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಡಾಕ್ಟರ್ ವನಿತಾ ಕೃಷ್ಣ ಸುಬ್ಬಯ್ಯರವರು ಬಯೋಟೆಕ್ನಾಲಜಿ ಅಂಡ್ ಇಟ್ಸ್ ಅಪ್ಲಿಕೇಶನ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು, ಸೊ ಮೇಡಮ್ ತಮ್ಮ ಬಗ್ಗೆ ಒಂದು ಕಿರು ಪರಿಚಯ.
Okay, I should introduce myself, sir.
No, Sudhagowda sir will introduce you. Please, madam.
Dr. Vanitha Krishna Subbaya has PhD Cancer Biology, DKFZ, Germany. Presently working as Assistant Professor, Goddess City University, Bangalore, India. And Madam published 14 research articles and many more postal presentations. Also, she has won ICGEB Postdoctoral Fellowship Award in 2009. Welcome, thank you. Over to you, ma'am.
Thank you, sir. Thank you very much for that kind introduction. A very good afternoon to all the students of PUC who are present for this webinar on the Science Talent Search Program organized by Karnataka Science and Technology Academy. I thank Dr. Ramesh, who has given me this opportunity to be a part of this program. My topic is going to be biotechnology and its applications. So I will try to do justice in terms of giving you the knowledge what I have gained a little bit in the past years in biotechnology. Although, as sir mentioned, my specialization is cancer biology, and definitely biotechnology plays an immense role in the cancer treatment these days. We will see it in the coming slides. So just to brush up the mind, I would like to give a little bit of a brief introduction to the field of biotechnology and how it exactly evolved to date. So as we all know, biotechnology can be defined in various ways in different countries by different scientists. But the bottom line is, it is a field which makes use of technology to convert any kind of raw or biological material into a useful product. And it can be from plants, it can be from animals, it can be from microbes, which act as biological factories, from which we are taking out certain raw materials or the products of these different species and converting it into something which is useful for the human mankind. So there is four major classifications of biotechnology, as you can see here. Medical biotechnology, also called the red biotechnology, which deals with improving the healthcare with the development of new therapies, diagnostic aids, and also treatment methods. We have the green biotechnology or the agricultural biotechnology, which mainly deals with increasing the yield for the farmers and better crop protection, because we all know that crops are always subjected to attack by insects and pests. And also how we can increase the nutritional values in the crops or cereals or any of it what we consume here. So then we have what is called as a white biotechnology or the industrial biotechnology, that is industrial scale production using biotechnological tools. So how can I produce massive amounts of a particular product, which is of use to the humankind, okay, at an industrial scale. That is what is industrial biotechnology dealing with. Then we have the marine biotechnology, or what is also called as a blue biotechnology, which is majorly focusing on improving the aquatic or the marine life, which is also important to humankind. So if some of the students ask me, what is it that is being taught under biotechnology, or what is it that I will learn? So I think this diagrammatic representation is giving you an overview of the fact that biotechnology is gaining the scientific knowledge through the various subjects which are being taught to students at the bachelor level, or even the master's level, or even the PUC students, which involves like biochemistry, microbiology, molecular biophysiology, immunology, genetics, etc. So the knowledge which you gain through these various subjects that you study during your curriculum is what leads to the development of biotechnological tools, which includes tissue engineering, cell and tissue culture knowledge, genetic engineering, DNA chip technology, or the DNA microarray technology. How do you produce monoclonal antibodies? It could be from plants, it could be from animals, and using antisense technology or the siRNA technology. Now, the most recent trend is the CRISPR-Cas9 technology. So the knowledge that you gain from the subjects leads to the development of these kind of biological tools, which in turn has its applications in the three broad areas, what I just mentioned, that is the medical, agricultural fields, for the various purposes which was mentioned, as well as the environment. Now, as I told you, briefly brushing upon the history of biotechnology, it can be divided into three broad phases. Ancient biotechnology, which was mostly based on some of the common observations which were made by people at that time, which was followed by classical biotechnology. And we have now currently what is called as the modern biotechnology. And the crux or the central importance given to modern biotechnology is through the recombinant DNA technology, or the genetic engineering, what we call it as. That is the main part of modern biotechnology. So coming to the stages of biotechnology development, I will just briefly go through these slides because I'm sure all of you have studied or learned somewhere in your textbooks about ancient biotechnology, which deals with the pre-1800 years, wherein the products such as cheese and curd were, the very first kind of direct products of biotechnology, as it was prepared by adding an enzyme what's called as rennet, which was found in the stomach of the calves. Then came the use of yeast, which is one of the oldest microbes that has been used for the fermentation in the industries for the production of it could be the bread, it could be the alcohol, and so on and so forth. And then we came across with the development of this mule, which is a crossbreed for the benefit of the humans. Mule is nothing but an offspring of the male donkey as well as a female horse. So we can see, or we can appreciate that though we know that modern biotechnology has taken us a long way, the people existing many years back were not bad, because they used their common sense and knowledge which was existing around to definitely come up with certain methodologies and products which could be useful for them at that time. So, and then we have discoveries of the cells by the scientist Hook. And then we have Antony van Leeuwen's Hook discovery of microorganisms by the scrapings that he took from the teeth. And from there onwards, we have the first development of the vaccination, which was shown by Edward Jenner. And we have the first enzyme, which was discovered and isolated in the year 1833, and the discovery of the viruses. So these are the kind of different discoveries which was done in the early periods, which I will not go in depth. I just wanted to show you how biotechnology has evolved as a field so that we can appreciate better. And in the year 1911, American pathologist Peyton Ross was the first person who discovered the first cancer-causing virus, or the tumor-causing virus. And the first virus that he discovered and showed that it can induce tumor in chickens was the Ross sarcoma virus. And also there was a discovery of phages, which are nothing but the bacteria which infect the viruses, or the bacterial viruses were discovered in the year 1915. Now you must be wondering, why is this picture highlighted so beautifully? It's because I have worked with viruses, and I really viruses, observing them under the microscope. So, and then we had the word biotechnology, which was first printed in the year 1919. And from there onwards, it goes on to the discovery of penicillin as the antibiotic, which is used for the disease treatments. Then we have the development of genetic engineering as a technique to transfer and select a piece of genetic material from one organism to the other organism. And of note is the fact that James Watson and Crick's discovery of the double helical nature of the nucleic acid, that is the DNA. And these are the few other discoveries which was done from 1968 to 1977. And then we had the development of modern biotechnology from 1945 to present, which involved the human growth hormone being synthesized for the first time, and smallpox, which was eradicated in the year 1920 after 20 years of mass vaccination effort. And then we have the development of the most important and the vital technique in biotechnology, I can say, by Gary Mullis in the year 1983, that is the polymerase chain reaction, wherein you're basically amplifying a small quantity of the nucleic acid, be it the DNA or even the RNA, into as many copies as possible so that it can enable the downstream processes. I do have a slide to show the overall procedure of our PCR works out in the forthcoming slides. Then we have the first pest-resistant corn and also the BT corn, which was produced in the year 1988. Then we have the development of banting, which is one of the extinguished species. And of course, not to forget is the fact that in the year 2001, the sequence of the human genome was for the first time published in Science and Nature, making it possible for researchers all over the world to begin developing treatments. Then we have the approval of Gleevec or the imatinib, which is a gene-targeted drug for the patients with chronic myeloid leukemia. I have one particular slide on that, which I will explain. And in the year 2006, FDA, that is the Food and Drug and this one, approved the recombinant vaccine, that is a Gardasil. Okay, it is one of the vaccines out of the two which is available as the prophylactic vaccines for cervical cancer. It is one of the first vaccines which was developed against the human papillomaviruses, which is an infection which basically infects the cervix in case of women. So women are majorly affected by this human papillomaviruses, leading to what is called as cervical cancer, and it was the first preventive cancer vaccine. So it is a prophylactic vaccine. So there is a difference between prophylactic vaccine and therapeutic vaccine. Prophylactic vaccine is the one which would prevent the onset of an infection, whereas therapeutic vaccine is the one which can treat a cancer which is already existing. So Gardasil 9 was one of them, which was, it is written as human papillomavirus 9-valent vaccine. Okay. 9-valent is because human papillomaviruses, there are 101 varieties or the species of the human papillomaviruses. Although cervical cancer is caused by two major viruses, that is the high-risk human papillomavirus 16 and 18. So we have the other categories such as the 6 and 11, which are considered as a low-risk category viruses, which is also known to mainly cause the oral cancers and the head and neck cancers. So basically, Gardasil was one of the first vaccines which is developed to act as a preventive measure for the cervical cancer, which is a very big burden on the world, because cervical cancer is standing, one among the top 5 cancers in the world. So I have to mention here that I had the opportunity to work with Professor Zulhausen, Harhard Zulhausen, who is the person awarded with the Nobel Prize in medicine, because he is the person who showed that human papillomavirus is a etiological or the causative agent for cervical cancer. So I had this honor to having worked under him as a PhD student. So that is where I developed my interest for the viruses. So, of course, what is lacking, still there is a literature gap in cervical cancer treatment, is because, as I told you, this is a prophylactic vaccine. But still there is a huge burden of women who are already suffering with cervical cancer. It can be the initial stages, SIN 1, SIN 2, SIN 3, or it can be the malignant stages. So we do not as yet have a treatment for those who are already infected and have reached the different stages. So there is still a lot of scope for research on cervical cancer in terms of developing the therapeutic vaccines, wherein now people are using immunoinformatics tools to come up with epitopes and all. So you students can always think about this as a field for future studies. So then this is one of the breakthrough events wherein this is one of the examples wherein they have developed the antibody that is the Gleevec. Was like a breakthrough in the cancer treatment. So many people believe that cancer is one such diseased condition that can never be treated. It's not so. There are a lot of cancers on which extensive research work is going on, wherein they are trying to develop drugs or even vaccines to combat it. So Gleevec is one such antibody which was developed especially for the CML, that is the cytomegalo, that is for the CML, this one disease condition. So how is this caused? Basically, this cancer, that is a myeloid leukemia. Okay, myeloid leukemia is caused because of the translocation between the two chromosomes as shown in this picture, that is a chromosome 9 and chromosome 22. Okay, which brings about a translocation between two of the genes, that is the BCR as well as ABL. So this particular figure on the right hand side, what it shows is that, so this is our BCR and ABL, which is basically a tyrosine kinase. Kinase are the enzymes which transfers a phosphate group, okay, and it takes the phosphate group from the ATP, adds it on to a substrate, which is an amino acid. In this case, it is the tyrosine. So what happens is when a tyrosine kinase, such as a BCR-ABL, transfers the phosphate group from this ATP to a particular amino acid, you are in a way activating. This is called as modifications to the substrates, right? So addition of phosphate group is a kind of modification. We have several other modifications like glycosylation, which takes place to proteins which are usually expressed at the cell membrane. Okay, so in this particular case that I'm showing here, phosphorylation of an amino acid can lead in turn to activation of cell signaling pathways, which will promote the cell proliferation. Okay, which will promote the cell proliferation, and as a result of which, in case of cancerous conditions, or in this condition, what we are seeing is happening is that there is an uncontrolled amplification leading to accumulation of the matured B lymphocytes. Okay, so what happens is this imatinib antibody, when how does it act? Imatinib is it basically goes and binds itself to this pocket where the ATP is present. As a result of which, what happens is phosphorylation of its substrate, in this case the tyrosine, as I said, is inhibited, because of which you do not induce the chronic myeloid leukemia case. So when there is an activation, there is a normal cell proliferation, but in conditions where there is overactivation, which leads to uncontrolled cell proliferation, it can be inhibited by development of certain antibodies. So imatinib is one such example, is a drug which was developed, which was a breakthrough in the cancer treatment, especially in case of the CML myeloid leukemia cases. So another group of scientists at Johns Hopkins, they have engineered mosquitoes that would resist the malarial parasite with combination of anti-parasite molecules. Now, this is something I think very amazing, because there was a period, or every time our season changes with a lot of rain and everything, people are suffering from this dengue so much that many people have lost their lives also. So it's so imaginative to think that we can develop engineered mosquitoes that can develop resistance to a malarial parasite, right? So so this is one of the studies which was done by the Johns Hopkins scientists, as I said, by Dimopoulos and his team, who have engineered the Anopheles mosquitoes with a combination of multiple anti-malarial effector proteins that could block the infection. So what is the strategy which they used is, they have used a single artificial gene, or what is called as a transgene, which encodes for 5 proteins that are known to kill the malarial parasites. What are those? It includes the Shiva1, which is a toxic from a spider venom, and you have the scorpine, which is a toxic from the scorpion venom again, and you have the melatonin, which is a toxin from the bee venom. So this is what I told you, that modern biotechnology is revolving around the recombinant DNA technology, which is nothing but you cutting out a piece of gene from an X organism or an X source, which you are interested and which has certain positive values or a good trait, let's say, and you insert it into any other organism or species so that that property is transmitted. So that is what they have done here. They have used a single artificial gene, what is called as a transgene, which will encode for 5 proteins, such as toxins. And being toxins, what happens is they help in killing the malarial parasites. So, as I was just mentioning in the beginning, CRISPR-Cas9 is one of the technologies which is used to knock out the expression of a particular gene. And so they have used that CRISPR-Cas9 technology to create the malaria-resistant mosquitoes also. One asks, is this really going to be successful in the natural wild environment? So this is something which the scientists are conducting studies on at the moment. So what they have done, whatever has been shown till now is at the laboratory setup, wherein they have engineered these mosquitoes, as I told, with the malaria-resistant mosquitoes have been generated. And how they foresee that it's going to work is by crossbreeding these kind of wild mosquitoes with those mosquitoes that is which are malaria-resistant versions. As a result of which, what happens is through the offsprings and through the generations, the resistant versions is what is going to become more seen. It is like survival of the fittest, and the process of evolution is such that only those which are efficient are survived, right? It's the same concept here as well. So this has not been tried on the wild environment, that is among humans as yet. It is a laboratory setup wherein they use the CRISPR-Cas9 mediated gene knockout of Anopheles gambiae, FREP1. Suppresses the malarial parasite infection. FREP1 is one particular gene which was shown to be important for the infection process, which they have knocked down using the CRISPR-Cas9. So moving on again to the specific applications in the excuse me, in the different sectors. For example, microbial biotechnology. So what do we mean by microbial biotechnology? It is making use of microorganisms, as we mentioned in the beginning. It could be the yeast or the bacteria, manipulating them and trying to create better enzymes. So now enzymes have got a very broad spectrum application in the industries. Excuse me, sorry. In the industries, in the in all the places, you know, enzymes have become very important, especially to mention one of them is the amylases, which helps in conversion of the starch. So one is you can use the microorganisms to create better enzymes, because enzymes are always required as catalyst to increase the activity of a signaling pathway or a biological pathway, for example. And they have also been used as efficient means of decontaminating the industrial waste products. It can be the oil spillage. I have a few pictures in the next slide. They can also be used to clone and produce large amounts of important proteins, which are used not only in human medicine, in various applications such as industries also. Because microbes, for example, if you take E. coli, most of the recombinant DNA technology, if you go through the contents, you realize that E. coli is one bacteria which they've used for many applications. It's simply because bacteria or the prokaryotic organisms are much easier to handle. They are cheaper, and you can bring about large production of, it could be enzymes or your desired proteins in a short period of time than using animal as a source or using plant as a source. Okay, so it is just the ease, it is cost effective. So in microbial biotechnology itself, we have seen the usage of microorganisms such as yeast and bacteria converting the carbohydrates such as starch and sugar into alcohol or acids. Now, some of the products which we use even till date through the fermentation process is the yogurt, the different kinds of flavored yogurts, the different flavors of the cheese, it could be the bread, it could be the wine, and fermented food products especially has shown to have immense benefits to us because of the fact that they increase the enzyme content, which in turn will help us absorb the nutrients in a better way, reducing the need for all the supplements which we tend to take most often these days. It could be the vitamin supplements or the protein supplements. So, you know, we are finding out natural sources by which how we can increase the, it could be the gut microbiota by simply taking the gut, which will increase the good microflora, which will help in the processing of my food that I eat. It could be the source of probiotics, as I just said, that is a source of good bacteria, which helps me restore a balance in the gut and helps in my digestion as well as in the immune health. So most often you must have seen when the small children, they get this stomachache, the doctors are suggesting just to give them curd. So this is the reason why we need to take curd, because it is a good source of good bacteria. And the lactic acid, which is created during the fermentation process, also kills E. coli, making it safer to consume than the raw vegetables. And the lacto-fermentation process stores food for longer. For example, if you take this pickles or even the kimchi, which is the Japanese favorite side food. So all these are the fermented versions of the food which are being used. Now, then other applications of microbial biotechnology is in terms of development of biofertilizers and biopesticides. Now, what is biofertilizer is, we know that the plants absolutely require phosphorus and nitrogen, all these things for the proper development or for the proper growth, for the tolerance of the adverse environmental conditions. Okay, so what happens is it is all freely available in the environment, but the plants are not able to use all this very efficiently from their ambience. As a result of which, what you do, you provide the microorganisms to the roots of the plants or to the seeds of the plants, which will help the plants absorb these kind of nutrients which are easily available to them in a much more efficient way and to grow better. So among them, it can be the rhizobium, which is growing in the nodules of the roots. Okay, so the nodules act as the factories where this rhizobium grow, which will help utilize the nitrogen which is present in a better way in the atmosphere. Or it could be the use of other bacteria as it is mentioned here, the cyanobacteria, clostridium, nitrobacter, okay, azobacter, all these kind of things are used as biofertilizers. That is, organisms being converted as fertilizers for the better development of the plants. Then we have the biopesticides. Okay, so biopesticides is trying to control the pests and the insects using, again, biological organisms, such as it could be microorganisms, it could be fungi, it could be viruses, to fight against these pesticides. So this is a table here basically which is giving you some of the examples of microbial pesticides such as strains of Bacillus thuringiensis. Then it could be the naturally occurring substances which acts as scented plant extracts. Okay, and it could be plant incorporated protectants. For example, the plant producing the Bt toxin. So these kind of biopesticides are extensively used in controlling the weed growth, as I have written here, because now weed is becoming a biggest challenge for the farmers because they're taking up most of the nutrients, the water that is available, everything for the actual plant growth. So it is inhibiting the plant growth in a way and reducing the outcome of the crops. So biopesticides is also now used for the weed control extensively. So under that you have bioherbicides, again, which is used for controlling the weeds. What happens when we use these kind of biofertilizers, bioherbicides, and biopesticides is that you are reducing extensively the use of chemical fertilizers or anything which is based on chemicals, okay, which is based on chemicals, because we know that addition of chemicals definitely might give me a better return to the farmers, you know, a faster yield. However, we should understand that we are compromising on the quality of the soil. Okay, we are compromising on the quality of soil, leading to perpetual deterioration of the soil properties, which is eventually going to affect the plants as well as the entire agricultural fields. So this is the reason why government of late has come up with a lot of initiatives wherein they are, freely installing these kind of vermicomposting units in many of the districts of Karnataka also. And even at Garden City University here, we have started up an initiative of vermicomposting, which is nothing but you're taking the naturally available products. It could be the food waste, it could be the green waste, and with the help of worms, that is the earthworms, okay, which are wonderful worm microorganisms, you can consider even them as, which will help you decompose all this waste into a useful product, that is my compost, with having good amount of CN ratio, phosphorus, nitrogen, everything that is required for the proper growth of the plant. So government is now in this need that, we stop the use of all these chemical fertilizers, chemical insecticides, pesticides, and we revert back to the natural sources that is our own microorganisms, which are very useful. Okay, and it is obviously environmental friendly. It is not harming the environment in any ways because it is from the natural source that we have been taking it. As I told you, we have fungal bioinsecticides. So fermentation technology is, so how exactly they use the fungi as a bioinsecticides is, fermentation technology will be used for the mass production of fungi. The spores will be collected or harvested and they will be applied to the insect-ridden fields. So when the spores are applied like this, the use of enzymes to break through the outer surface of the insect's body. So that is the molecular mechanism how these fungal bioinsecticides work. So as I was just mentioning, we have Bt cotton, which is nothing but the Bacillus thuringiensis cotton, which is one of the first crop protection products from biotechnology. So this is very simple. Bacillus thuringiensis is nothing but again a soil bacterium, which produces a kind of toxins, okay, what are called as a cryproteins, which are like in the crystals. So these can act or it can kill directly the pests, especially the bollworms. One variety of the worms which is known to be very harmful to the cotton, which has reduced the production of cotton to a very large extent, causing taking a big toll on the farmers, is these bollworms, and along with the other worms, of course. So what they do is they take the cotton cells and they take the promoter of this Bt gene, that is a Bacillus thuringiensis gene, and turn on the production of the prototoxin as shown in the figure right here. So the promoter will help in the production of prototoxin in the plant tissue. So when the insect is eating that plant tissue, the gut enzymes of the insects itself will activate this Bt toxin from the prototoxin to the toxin version. Okay, so it will be converted and this toxin in turn will eventually lead to the death of the pests. So that is one of the examples of biopesticides we can call, wherein you have used Bacillus thuringiensis, which is a soil bacterium, taken out a gene from it, which is known to encode for the cryprotein or the toxins or the endotoxins. Now that endotoxin is introduced into the plants of the cotton, so it is in a way a transgenic plant. Okay, so Bt cotton. Now, the three protein primary components which was used in as a genetic package, what we call as a cassette, a gene cassette, which was inserted into the cotton plant, was the protein gene, of course, the Bacillus thuringiensis gene, which was modified for improved expression in the cotton. So when we mean improved expression, we want excess production of that particular gene. Okay, so that is enhanced expression of the gene. And then a promoter, of course. So people must be aware of the fact that the expression of a particular gene is driven by what is called as a promoter region upstream of a particular gene. So this promoter is something which I can decide to put a weak promoter or a strong promoter. So you give in a promoter and then next to it you have the gene which is going to be activated. In this case, it is the Bt gene. And of course, you have what is called as a genetic marker. Okay, a marker is always required because you want to select those transgenic plants which has taken up your foreign gene of interest. Okay, so this is the three simple components which was put as one genetic package or a gene cassette, and it was transformed or it was introduced into the plants. So this is, for example, the slide which I was telling you, wherein the microbes have been used in the bioremediation. That is, acts as a remedy to clean up all the soil contaminations, groundwater purifications, Okay, so these are some of the pictures which shows where and all it is being used, the microbes. Then moving on to agricultural biotechnology, it is how can I improve the crop plants, okay, using the genetically modified plants, okay, again by the same technology of recombinant DNA technology. How can I improve the tolerance of plants or the crops to the herbicides? How can I increase the resistance to the pests, okay? And drought resistance, nitrogen fixing ability, and acidity and salinity tolerance. So these are some of the major objectives as to why we as biotechnologists would want to modify the plants. One is to bring about the crop improvement in the following categories which I mentioned. And the second objective of biotechnologists, especially the agriculturists, would be how can I increase the nutritional value of the crops? Because we know that, there is a lot of deficiency. We feel that we have been eating enough everything, but still we are succumb to a lot of infections, especially these days, the common flu type of things, cold, cough, fever has become so fine. But if I go to think about it, I feel that I'm eating enough, but still there is some problem. That is because either whatever we've been eating is not being absorbed in sufficient quantities that is required by body, or the quantity is less. So how can we improve the natural value of the crops? It could be the rice, it could be the ragi, or whatever that I'm eating. How can I improve the quality and safety of that? How can I reduce the saturated fatty acid contents in the vegetable oils that I'm taking? So all these are certain aspects which they address through the agricultural biotechnology. And one example which I have mentioned in the future slides is about how golden rice was developed for the enhancement of the beta carotene content. Yeah, because many of the kids, especially, they have a lot of this vitamin A deficiency, which will eventually lead to problems with the eyesight and leading to blindness. And then in agricultural biotechnology, one more most important application is with respect to plantibodies. Plantibodies, in simple sentence, we have to define means it's nothing but antibodies which are produced using the plant as a host system. So we must have heard of antibodies which are produced from animals. Yeah. So those are the recombinant antibodies. So first of all, how did the concept of recombinant antibodies come into picture is, we know that when there is an infection, let's say I'm infected by a pathogen. So what happens is the humoral immune response of my body gets activated, because of which the B lymphocytes start generating what are called as antibodies. Now, these antibodies, they will go and bind to specific epitopes, or what we call as antigens, which are present on this pathogens. So if there is a wrong antibody which is produced against the antigen, it can lead to infection and further complications. But what was seen is that we can generate something called as recombinant antibodies just by fusing two genes, two parts of an antibody. So antibody basically, I have a picture here in one of the slides, which I can show. Yeah, so in this picture, if you see, this is like an antibody structure wherein we have the heavy chain, the light chain, and then we have the constant region, and we have the antigen binding site. So this is the site where the epitope of an antigen or a pathogen would bind, and this is the constant region. So what scientists realize is that by fusing this constant region from a human and the binding region from that of a mouse, you can create what is called as a recombinant antibody. So in earlier days, this recombinant antibody production was happening using mouse models. Okay, so of course it is good, but then it comes with a lot of challenges, because handling animals is not easy, or mammalian cells is not easy. It's not cost effective for us. It takes a lot to maintain these kind of animal models as well as cells, and etc. And it can also be accompanied with certain pathogens, which can actually in turn do harm to me instead of doing good. It can elicit a immune response which is not good for me. On the other hand, what happened is people have reverted to using plants as the bioreactors, okay, for the synthesis of the same desired antibodies what we used to produce in animals. Now, it is easy, right? Because plants are not going to cost us too much, and they don't have, they can be grown easily. So it becomes cost effective. It is not coming with any additional pathogens or additional immune responses. So plantibodies have become, one of the biggest booms again in biotechnology. So this golden rise is what I was talking about, where in agricultural biotechnology, one thing is increasing the nutritional value of the crops, for example. So in this case, as I told, vitamin A deficiency related disorders could be overcome by using the gold rice, okay, which is based on simple principle that rice plants acts as a factory themselves to synthesize the beta carotene, okay, and this could be which is fully active in the leaves or even in the parts of the grain as well. And this was done by just adding only two genes. One is the plant phytoene synthase, PSY, or the bacterial phytoene desaturase, what is called as in short the CRTN. Okay, so this ensured the insertion of these two genes ensured that the pathway is turned back on and beta carotene is successfully getting accumulated in the grain, which is required for the good health. Then the technologies which is implied in plant biotechnology would, as I told you, involves genetic engineering, tissue culture, the molecular breeding process. Now, then there is another big aspect of agricultural biotechnology, that is the genetically engineered crops or the modified crops. First genetically modified plant in the lab, okay, in the lab setup was the tobacco. Then there is also there are a lot of examples. There are a few which I have mentioned here. Tomato was the first commercially grown genetically modified whole food crop, which was modified in such a way that it could ripen without softening. And there are 5 major categories of the crops, 2 of them which is very much resistant to the insects or to insects and herbicides, that is a corn, as well as the genetically modified corn and genetically modified the cotton, as we just saw the BT cotton. And the other crops includes the soybean, canola, as well as the sugar beet, which are also having shown to develop the resistance to herbicides. Now, how do you produce a genetically modified crops? In simple steps, if we have to understand, you will first identify and locate the gene for the plant traits that is required, then designing the genes for the insertion. But once it is inserted, it has to be cloned into a bacterial vector. It has to undergo modifications before it is properly effectively inserted into a plant. And then you would insert that into a plant, that is called as transformation. So you would transform the plants. Now, transformation of the plants can be done through the gene gun method, or it can be done through one of the most popular methods using the Agrobacterium tumefaciens mediated plasmid. So Agrobacterium tumefaciens is again another soil bacterium which is known to cause the crown gall disease in case of the plants. So this it does with the help of a plasmid, what is called as tumor inducing or the TI plasmid. So what they did is they took out all the cancer causing components from this TI plasmid and you simply replace it with the genes of interest that you are interested in. Okay, with certain other components which is required for it, of course. I will not go into the details of it. So tDNA of the TI plasmid is what is very important for this process. So you insert your genes with the left and the right borders of the DNA, and then you make this plasmid, introduce basically this plasmid through the Agrobacterium tumefaciens into the plants. And then you, of course, select for the positive plant, which is taken up a foreign gene of interest. And then you regenerate the whole plants under controlled environmental conditions in a series of media containing nutrients and hormones, what is called as through a process what is called as tissue culture. So this is another example wherein a lot of anti-cancerous drugs have been produced through the plant-derived products. Okay, microbial production of plant-derived anti-cancer drugs, because cancer is one field which has got a lot of space to work with. So there is a lot of continuous research which is being happening all the time. Animal biotechnology is another aspect wherein we know that animals act as a source of medically valuable proteins. As I was just explaining how they can be used, mouse models are used for the production of antibodies. Transgenic animals, there are many reasons why I would want to make a transgenic mice model. So in order to study how a gene functions or how what is the pathway which is followed by a particular disease causing gene, or in order to study the whole disease itself, right from starting stage till the ending stage. As I told you, for example, cervical cancer. I cannot possibly take a human being and I can study it, right? So I will make a mouse model by infecting the certain regions of the virus and seeing that, okay, what kind of tumor will it first cause? The solid tumors, and then how it progresses from SIN1 to SIN2, SIN3, and then the final invasive stage. So I can create models like this. Our transgenic animals can also mean wherein I am putting in the gene of interest to increase the milk production. Okay, this is very commonly seen. So you can increase the production of the milk, or you can use it to produce certain antibodies. Okay. Dolly also, for example, that was a clone. Okay, so that is also a transgenic animal. And animals are also important models for the basic research, like the gene knockout experiments, designing and testing of drugs and genetic therapies. As I just mentioned, not all experiments or not any experiment is possible unless we reach the clinical trial stage. For a particular treatment of a particular disease. So usually the scientists are doing experiments at the cell line level, and then they are using an animal model, such as it could be the mouse, which is the closest relative to the humans. So you can design, animals are important models for such things. So you can do gene knockout experiments. Knockout is nothing but you're trying to suppress. Knockout is complete removal of a gene. So, you're completely removing the gene, so the function is dead. But you can also do something called as siRNA technology, mRNA technology, as I was just telling you, using what is called a small interfering RNA fragments, wherein you are only suppressing the mRNA expression levels, which will affect the protein productions. But you're not completely abolishing the existence of the gene and the corresponding function. You're only silencing the expression transiently. But if you use short hairpin RNA technology, that is the shRNA, to completely abolish the expression. Animal cloning, the Dolly, for example, what I told. So this is picture where I said that, transgenic animals can be used for large-scale production of therapeutic. It could be proteins. It could be for increased milk production. Okay, it could be production of clotting proteins. Gene knockout is, as I just told, you are disrupting a gene in the animal, and you're trying to look at what are the downstream functions which is affected because I have disrupted that particular gene. As a result of which, it helps me understand what is exactly the function of that particular gene. Now, forensic biotechnology is another field which is of immense use to humankind through many of the techniques. One of it is the DNA fingerprinting technique, okay, which enables one to understand who is the culprit in a crime scene, who exactly committed the crime. It helps me identify the paternity, okay, and identification of human remains, endangered species can be identified, and also tracking and confirmation of spread of the disease. So in short, if I have to tell how does DNA fingerprinting work, is the principle is very simple. So we know that almost all of us are identical in terms of 99% of the gene composition, except for 0.1%. That 0.1% variability between two individuals is because of the existence of what are called as short tandem repeats, okay, or the variable tandem repeats. So these are nothing but the tandem repeats of short DNA sequences, which is unique to a person. So in this DNA fingerprinting, what they're trying to do is they are trying to analyze these kind of differences in these short repeated sequences between two individuals. So the process is very simple. Whereas, for example, there is a crime scene. And as you can see in this left figure here, what is pointed out is that one blood sample. I take the blood sample, what is seen at the crime scene. I isolate the DNA from that particular blood sample. The DNA will be fragmented through the restriction enzyme digestion. Okay, because it's not feasible for us to read the entire sequence if it is in the full genomic DNA. It's very difficult. So even for the sequencing purposes also, what we are doing is we are fragmenting the DNA into short pieces, which is workable, easy to handle. So you fragment it and then you run it. You separate the fragments which are cut on a gel, which will separate out based upon the molecular weight. And these fragments, after being separated, they will be transferred to a nylon membrane through a technique of what is called as southern blotting. And then you probe it using specific, you hybridize, you do the process of hybridization, wherein you provide a DNA probe. Probe is nothing but a short stretch of, again, DNA sequence, which is complementary to any of the sequences which is present in the genomic sequence. Okay. So based upon the hybridization process, what happens is, after washing all the unbound stuffs, I don't want to go into the details of the southern blotting technique and all, but I am trying to give you the idea that the DNA is, the total DNA will be isolated from the blood sample. It will be cut into small pieces through the use of restriction enzymes. Those cut pieces will be run on the gel. Now, obviously, I want to transfer the bands from a gel onto some substrate which is readable for me. That is nothing but my nylon membrane. Now, then I do the process of probing it with the DNA probes, and which will be detected through the X-ray film, okay, through the autoradiography method. So I know what is the probe that I have used, okay, with what kind of sequences that I have used. So based upon that specificity, I will be able to make out the pattern. So if you see this is basically a nylon membrane, and this is the fragments which is being transferred from the gel, okay. So if you see, this is my marker lane, you can ignore this. So these are various examples. So how you learn it is, you start comparing each lane by lane. For example, 1, 2, 3, 4, you start comparing to see the band patterns. Okay. So you can see that across the samples, maybe this band is very unique. Okay. And so like this, you compare the pattern across the lanes to see which is closest to the to the blood sample, what is found on the crime scene. Okay. So for example, if this is the sample of the blood which was taken from the crime scene, and these are all my suspects. Okay. These are all the suspects. So you compare across and you see which is matching, okay, to the blood sample that was collected from the region of the crime. So that is how the DNA fingerprinting works. And for the DNA fingerprinting, as I told you just now, you will be isolating the DNA from that blood sample. So sometimes what happens is, you don't have liters of blood. You just have some small 5 ml or 2 ml or 1 ml of it. So you use a technique, what is called as polymerase chain reaction, which I'm sure you've already come across, to amplify, to make many copies of that particular DNA. So the simple procedure of the polymerase chain reaction will be, if these two bands you consider as my double-stranded DNA, you have to first denature it, that is, separate out the 2 strands. After separating it out, you provide it with what are called as primers. Now, primers are required because the next step, which is nothing but addition of the free nucleotides to these primers, is mediated by DNA polymerase enzyme, and DNA polymerase enzyme does not initiate the synthesis. It requires an already existing primer. This block here, for example, is my forward primer. This is my reverse primer. So the 2 strands will be first denatured by heating it at around 95 degrees Celsius for a few seconds. So after you separate them, you add the remaining components such as the primers, the free DNTPs, that is nucleotide triphosphates, and then you provide the enzymes such as a DNA polymerase enzyme. Okay, all these components you add. So what is happening in the second step is, primers will go and adhere to the complementary site on both the strands, and then the primers will be extended based upon the Watson and Crick model of complementarity of the bases with the help of the enzyme DNA polymerase. So as a result of which, the one set of copy which you had 2 strands of DNA will end up giving you 4 strands of DNA in the cycle 2. Okay. So in cycle 1, you started off with just 1 double helical DNA. And after cycle 2, that is elongation of the primers by the addition of free nucleotides, you will end up getting 4 copies. So like this, you set the number of copies in the PCR cycle. It can be 20 cycles, 25 cycles. You usually don't go for more than 25 to 30, because then you'll end up getting all non-specific products and the primer dimers. So up to 25 cycles, you'll be able to produce a lot of that initial DNA, which was present in maybe just in 5 copies. You can produce it in 1,000 copies, and that will help you do the southern blotting for the DNA fingerprinting. And polymerase chain reaction, as I told you, is one of the art techniques or the core techniques of recombinant DNA technology. A modified version of this polymerase chain reaction, that is the real-time quantitative PCR, is what is being used during the corona pandemic that we had to quantify the viral load that each individual was infected within. So, for example, if you see this is a gel here, wherein you have the defendant's blood and the sample which was taken from the genes, blood from the defendant's clothes, and this is the victim's blood. And you can see that these are the DNA band patterns which are seen. So you can very clearly see, similar kind of patterns, okay, among the three individuals. So this is how the forensics field used to decide who's the crime. And medical biotechnology is also involved in other applications such as preventive medicine, diagnosis of health and illness, treatment of human diseases. And medical biotechnology also involved a very huge project that is a human genome project, wherein they have come up with the sequence of the entire human genome. And this in turn is going to help us in targeted gene therapy, because I will know the sequence, I will know the genes which is encoded by it. So having analyzed that, okay, X particular gene is known to cause this disease, I can try to target that particular gene in the gene therapy. Then we have stem cell technologies, because stem cells can be grown in the lab, although this is a very challenging field. So it can be grown in the lab, then treated with different chemicals to allow them to develop into specific kinds of tissues, which can be used for transplantation purposes. And the current use of the stem cells, as some of them, not all of, I've just mentioned a few, is being used for diabetes as well as the spinal cord injuries. So how will medical biotechnology change our lives in the years ahead means, so all the information that we have gained from the Human Genome Project will definitely give us a lot of insights into the function of the human genes and what are the other controlling factors that regulates the expression of these genes and the functions of the genes. Because it is not enough if I just know, okay, this particular sequence corresponds to this gene. I also want to understand how this particular gene is being regulated at the transcript level, at the protein level, all the epigenetic changes which is happening. So all this kind of information is achieved only when I know the sequence of the genome. Okay. So then the medical biotechnology also has helped us a lot through the analysis of the single nucleotide polymorphisms. That is the changes or the mutations that we see in the DNA sequence from one individual to another individual. The VNTRs, which I just spoke of in the DNA fingerprinting, is also similar to this. That is the changes in the tandem repeats of DNA between two individuals. Now, these kind of variations are also shown to cause some of the genetic diseases such as the sickle cell anemia. So these SNPs will help one identify genes which could be involved in different medical conditions such as arthritis, stroke, it can be cancer, heart disease, diabetes, and other related cancers. For example, to quote one, SNPs in breast cancer, the single nucleotide polymorphisms in case of BRCA1 and BRCA2 genes, which is mainly involved in breast cancer, has been identified and shown to promote the breast cancer. Now, when you identify these kind of SNPs in the BRCA1 and the BRCA2 genes, obviously the doctors or the pharmaceutical companies can come up with a better treatment for the people, focusing only on those specific SNPs which have contributed a lot in promoting the breast cancer. Then some of the challenges of the 21st century is how can we benefit from the Human Genome Project? Because when we know the individual's human genome sequences, we can also identify the SNPs, as I just said, the single nucleotide polymorphisms, and a particular change sometimes, for example, as you can see in this figure from person 1 to person 2, there has been a single nucleotide polymorphism, that is a change from the guanidine to the thymine residue. But it has not changed in the the structure of the protein that it produces. So it is still a normal protein. So some of the DNA variations have no negative effects on protein structure or function. It's not that every mutation which we see will definitely have an effect on the protein. So in this, from person 1 to person 2, the SNP has not brought about any change. However, you can see that from person 2 to person 3, it is changed from T to adenine again, which is resulted in low or non-functioning proteins. So there can be other variations variations which, can lead to a genetic disease or increase susceptibility to a disease, depending upon the kind of mutation and what is its importance. So how can one test a person's DNA from many different SNPs? You can use what is called as the microarray technology. So in brief, if I have to explain microarray technology, it is used in various applications. One of the important areas where DNA microarray can help me is in the cancer field, wherein, as you see in this picture, it is basically helped me quantify the RNA expression, okay, both in this case, for example, a normal patient and in the tumor patient. So you isolate the total RNA, okay, convert it into cDNA, that is a complementary cDNA using reverse transcriptase enzyme, and then you will add that cDNA onto a chip, okay. So these two slides, what you can see is nothing but a DNA microarray chip, which has got thousands of genes on it, okay. It can be the cDNAs or it can be oligonucleotide microarrays. So depending on what you choose, you add that cDNA, which is derived from the RNA from these two samples, onto the different spots, the 1,000+ spots which is present on the microarray chip, and based upon the color that it elicits, because the RNA, for example, from this sample and this sample will be usually labeled. In this case, it is labeled with biotin. So you can also label it with different kind of color dyes, okay, for example, Psi3, Psi5, so that there is green and yellow light which is being emitted, signal, sorry. So based upon the differential labeling, okay, you will be able to see which is the subset of genes that is either upregulated or downregulated or has no changes in the gene expression level. Okay, so for example, tumor cells in comparison to the normal cells, you have a subset of genes which turns out as red spots. Okay, so those red spots are the ones. Which are showing the upregulated genes. Means the expression level of that particular gene is very high in that tumor patient. So this information that, okay, this subset of gene is upregulated, contributing to the cancer, is a lot of information for the people to develop drug or any kind of therapy against that particular tumor. Okay, so DNA microarray technology, in short, is a technology which helps me quantify the amount of transcript or the mRNA which is present in a normal patient versus the tumor patient, in this particular example that I'm talking about. Okay. Then how will medical technology change our lives in the years ahead? We have other fields like pharmacogenomics, which is nothing but customized medicine. So, the idea is that in future, people will develop what is called as personalized medicine. Okay, so that is based upon the gene sequence of me, for example, and the kind of mutations or SNPs and the changes that my gene sequence has got. They will develop customized medication, okay, for each individual. Because most often we have seen one medicine which works on one person does not have the same effect on another person. So personalized medication, okay, so this can be achieved only through a lot of work, that is sequencing and learning the expression levels of different genes in my body, etc. So there you can use techniques like DNA sequencing, genome sequencing, DNA microanalysis, etc. Then we have metabolomics, which is basically a study of the metabolites. Metabolites come at the end. It is like, the gene sequencing, then you have the RNA level, then you have the proteins, and then you have the metabolites. So metabolomics is nothing but the study of those small, even 1,500 Dalton molecules can be studied through the process of metabolomics. So also we have the nanotechnology which is coming up, wherein nanotechnology involves very small devices to deliver drugs, or especially in case of cancer, a lot of research is being going on, wherein one of the problems with the drug treatment in case of cancer or any other diseases is the off-target effects that it has. That is, it is not localized to the organ which is actually bearing the viral infection or bacterial infection or the disease, but it goes and affects the other parts of the body, because of which you have a lot of side effects. So how can we minimize this is by targeted therapies. That is, it should go and affect only the region which has got affected in my body. So for that, they are developing these kind of lipid-based nanoparticles which will carry the therapeutic drugs, and research is going on to see how it can be delivered to the required spot, not affecting the surrounding tissues. So gene therapy is what I was speaking about also, how targeted gene therapy can help us, and regenerative medicine, genetically, wherein you use genetically modified stem cells of patients to treat genetic conditions. Okay, in future, scientists will be able to isolate the adult stem cells from a patient with a particular genetic disorder, manipulate these cells by the gene therapy approaches, and reinsert the cells into the same patient to help treat their genetic diseases. So in terms of the job opportunities and everything for biotechnology field, this is the graph which basically shows the number of biotechnology companies that has been coming up, generating more than $63 billion revenue worldwide. Okay, so the increase in the number of pharmaceutical companies and everything shows the importance of biotechnology. So coming to jobs in biotechnology, of course, research and development is always there. It is a field which is very absolutely essential, both at the preclinical level. It could be the discovery research, bioinformatics, lab safety, very basic science also. If there was no basic science research happening, today the vaccination for coronavirus couldn't have been possible, I think. So, you know, research and development is like the heart of biotechnology, one of the jobs. And you can also be in the manufacturing section, the QC section, that is a quality control, and the clinical research, data management, for example, consultation, finance and administration, business development. See, because in the legal especially, if there is some issue which has happened in a pharmaceutical country, I mean, in a pharmaceutical sector, a lawyer without a biological background cannot do much justice. So a lawyer having a biotechnology course can do better justice in that particular situation, especially if it is a havoc which has happened in pharmaceutical company or in a life science sector. So the best approach to landing into a good job in a biotech company is obtaining degree. It could be in any of the subjects. As we learned, it's an integration of all the subjects leading to the field of biotechnology. So it could be molecular biology, biochemistry, bioinformatics, minor in information technology, taking a lot of math courses, having great skill in oral and written communication, having collaborative teamwork, and it also pays you well, and it is personally rewarding field. So these are some of the good jobs that you can find in biotechnology companies. Now, these are some of the slides which I had prepared, which I think you already would have read in your course curriculums, like what are the major tools of recombinant DNA technology. That is use of restriction endonucleases, which are nothing but enzymes which cut at specific sites in the DNA. So how is this entire recombinant DNA technology possible is because I have certain set of enzymes, what are called as restriction endonucleases, which is able to cut at particular sites that I choose. Okay, and after cutting, you need to, there are different types of restriction enzymes. So after cutting the restriction enzyme, you need to insert it or ligate it along with the vector backbone. Okay, so what is the certain features of the vector backbones, if you ask me? The vectors should be small in size. They must be easy to manipulate. It should be easy to transfer from cell to cell, easy to isolate from the host organisms. And some of the common features, what the vector should have is, it should have an origin of replication, because you want to amplify the gene of interest of yours, which is carried by that particular vector. It should have a reporter gene, which is nothing but the genetic marker, which we spoke about in the previous slide, a promoter to drive the expression of that particular gene, an antibiotic resistance gene, which also acts as a marker. Okay, and it should have a cloning site, that is a multiple cloning site, into which you will be inserting the gene of interest. And all these other things like the epitopes and protein purification tags finds its application in other fields like the affinity purification. That is wherein I want to specifically purify one particular protein. Okay, so that time you can add epitopes or any of these GST tags, GFP tags, which will help you in protein purification. But some of the basic components of a good vector would be the ones which it should have: origin of replication, a promoter, an antibiotic resistance gene, okay, and a multiple cloning site for sure. And these are the different types of vectors which are used in molecular biology. The plasmid is the one which is easygoing, very easy to handle with, okay, most often used for the DNA manipulation. And then from there on, many other vectors have evolved. like the lambda phage based vectors. We have the gossamins, parchments, N13, the bacterial artificial chromosomes or the yeast artificial chromosomes, and so on and so forth. Now, why it evolved from plasmid to so many vectors is because of the need to insert genes of bigger sizes. So if you can see this column here, maximum insert size in terms of kilo base pairs, what is possible in a plasmid is very minimal, 10 to 20 kb. Okay. So, but if you want to do genome sequences with very big fragments of the DNA, which is running up to like more than like 1,000, 2,000 kbs and all, you obviously want to use a better vector which can take a larger piece of fragment. So that is the reason why there was an evolution from plasmid to all these other different kind of vectors. So I think I would stop at this. And I would like to thank Karnataka Science and Technology Academy once again, along with the organizers who gave me this opportunity to share some views on applications of biotechnology with our dear students. And 2 questions based on my presentation will be mailed to you through the organizers. Thank you all for your attention.
Thank you very much, Dr. Mrna Technology. It's a wonderful presentation on biotechnology and its applications. Now I request participants, if there are any questions, please mention in the chat box. I think there are no questions in the chat box as of now. So, sir, I request our honorable member of Academy, Dr. Ambekar Eknath, sir, if there are any questions or any feedback, sir, please. Sir.
Hello,
Hello, sir.
can you hear me?
Yes, sir. Good afternoon.
I think
sir.
you cannot see me, is it?
No, sir, I cannot see you, sir.
I'm not able to get the camera on. But anyway, first of all, on behalf of KSTA, we would like to thank you, Dr. Vanita, for a wonderful lecture. It was really wonderful because, you know, it's a very complex and a very diverse subject, which has been explained in such a simpler term and covering almost everything and keeping our attention intact throughout. Thank you very much, madam.
Thank you, sir.
Wonderful,
Thank you, sir.
wonderful job. There are only 2 things I would like to comment on.
Sure, sir.
Because as a matter of history, basically, all the things which you explained, all these developments and advances and everything has happened during the past very, very short span of time of just 3 to 4 decades. That's it. It's a wonderful subject which has just taken off.
Yes, sir.
But at the same time, it has also had its problems too. You know, for example, there was a lot of resistance, especially on transgenics in the beginning stages. The word transgenics was a taboo. And there are, even to this day, there are some platforms who are consistently and very vehemently, they are opposing biotechnology, especially
Yes.
genetic GM crops.
Yes, sir.
You may recall fields where the BT cotton was being grown, they had burnt those cotton.
Yes, sir. Yes, sir.
And a little bit of that touch up would have given a very, but anyway, it does not matter. Now I do not know what is the status now. Do we still have that vigilante type of people?
No, sir. So I think from my, if I may interrupt you, I think in other countries, like in some parts of US and all, so this kind of resistance, what you said, has been overcome, wherein the government is promoting, you know, this kind of transgenic crop, I mean, genetically modified crops, and even for the golden rice also. I think the people have started slowly accepting it. This is my opinion from whatever.
Yeah, usually there is a lag period between the technology being introduced and the acceptance by a society, you know, that we know. But within this 40 years, that is what I really admire about this particular subject. and it is such an exciting field and so much to do,
Yeah.
and
Yes.
all for the welfare of the human beings and the animals and other things which we would talk about. So once again, thank you, Dr. Vanitha Krishna, for
Thank you, sir.
a
Thank you,
wonderful introduction to a very interesting topic. And I hope the students here will follow your steps, and we can see that probably biotechnology is the area where you might be expecting most of the Nobel prizes in the future than
Yeah,
pure
yes, sir.
subjects like this. Thank you very
Yes.
much, madam.
Thank you. Thank you. Thank you very much, sir, for your kind words. Thank you very much.
Thank you very much, sir. Now, I request all the participants, please do ask questions if you have any questions. Otherwise, you can mention the questions in the chat box now or later in the WhatsApp group or through email. You can contact us so that we can get back to Dr. Mrna Krishna and revert you with the answers. As of now, I would like to wind up the session, today's session, with formal vote of thanks. First and foremost, I would like to thank Dr. Mrna Krishna for accepting our invitation and delivering this today's session. Thank you very much, Dr. Mrna, on behalf of KSTA and on behalf of all the participants and our honorable members and other students and teachers who have joined with us in this YouTube platform as well as Cisco platform. Thank you very much. Also, I would like to thank our chairman, Professor S. Ayappan sir, and our honorable members today who have joined with us, Dr. Ambedkar Eknath sir, and other colleagues, Sri Degoda, and the technical key person, Anton Lobo, who is the main person behind this. And also, I would like to thank our CEO, Dr. A.M. sir, and other technical and non-technical team. Last but not least, I would like to thank today's participants, who are not only the students who are selected in this National Talent Search program. Apart from them, there are many teachers who are doing this program continuously, either in YouTube or Cisco platform. The teachers and science aspirants, general public also following these talks in YouTube, and they are giving us commendable feedback to continue. And there are only requests for changing of timings of the sessions. So the more number of students will be able to join. In the future, we are planning to suitable timings for them also to increase the audience for this talent search program. With this, thank you once again. Thank you very much to one and all. Thank you, madam.
Thank you, sir. Thank you so much to everybody.
Thank you, sir.
Thank you.
We will send the questions to WhatsApp group. WhatsApp group of this by Sri Degoda will be communicating, and you can send the answers. We'll
Sure,
send you the answers, ma'am. You can choose the best two answers. We will be awarding them cash prize.
Sure, sir. Thank you, sir. Thank you very much.
Thank you very much. Thank you.